Hi, my name is Jesse McCane. I'm the data coordinator for the Eagle Fish Genetics Lab, and the title of my talk is FishGen.net, a final repository for salmon and steelhead genetic data as part of GSI and PBT projects in the Columbia River Basin. So, it's becoming more and more understood that genetic data is one of the keys to successful fish management. More and more state, federal, and tribal agencies are turning towards genetics in order to help address various fish management problems. Genetic data is already being used for things like marking hatchery stocks, conducting hatchery evaluations and reform, conducting fishery evaluations and assessments, and developing and implementing harvest plans. Now, these are just a few ways that genetic data are already being used as a tool for fisheries management. To give a more specific example, over the last several years, the Pacific Coast Salmon Recovery Fund has contributed funding towards a parentage-based tagging program in the Snake River Basin. For those unfamiliar with PBT, this involves genotyping all adults spawned at various hatcheries, which in turn allows us to identify offspring at any age from these parents and assign them back to stock, hatchery, and even rearing location. PBT has already been used to do things like assessing straying of hatchery fish, assessing B-run stock composition in Zone 6 fisheries, assessing hatchery origin of Kelt steelhead migrating past Lower Granite Dam, and assessing the stock composition of steelhead and Chinook returning to the Snake River Basin. So for this project alone, we've genotyped over 47,000 steelhead and 66,000 Chinook salmon over the last 7 years, and that's just for the Snake River Basin. So outside of just our labs, various state, federal, tribal, and university labs are generating hundreds of thousands of genotypes every year in order to meet these information needs. Needless to say, the costs involved with generating these data are substantial, and the sharing of data between labs is crucial in order to reduce costs and workloads. So this in turn requires some common data standards in order to make the sharing of data more streamlined. So that brings us to this concept of a shared online repository for genetic data. Now, a standardized online multi-agency database would simplify the data sharing process, which would in turn increase efficiency and decrease the costs normally involved in sharing that data. A genetic database repository could also help in meeting reporting requirements for various funding agencies. Although most fish genetic labs have their own in-house database for storing and managing data produced in their own lab, none are really suitable to fit the purpose of a long-term shared data repository. In 2012, we became interested in the construction of a web-based GIS interface database that would serve as an all-in-one destination for genetic data from large-scale steelhead and salmon projects on the Pacific Coast. Not only would this database serve the immediate needs of our department and collaborators, but it would also be built in such a way that it could accommodate data from other geographic regions, species, and genetic markers from labs across the Pacific Northwest. Now, it's important to note that we aren't the first ones to do this. The GAPS and SPAN databases were two large collaborative efforts to build online repositories for Chinook and steelhead genetics, respectively. Both of these databases successfully accomplished what they set out to achieve. However, our goal was to build a more versatile database that would accommodate a wider variety of species and marker types. So let's address two important questions: who built it and who paid for it? We contracted the building of FishGen to a company called Resource Data Incorporated. They're a software development company that specializes in system integration and geographic information systems. They were an appealing choice for this project as they have experience working on many other fisheries-related projects such as AK fin, pack fin, e-landings, and the Yukon River fish tracking system. RDI also has local offices in Boise, which helped facilitate regular project meetings. For funding, we applied for and received $250,000 from the Pacific Coast Salmon Recovery Fund and the Bonneville Power Administration. So let's take a quick tour through FishGen.net, where I'll give a quick overview of its features. So this is the homepage. There's a menu bar across the top. We've got our collaborating agencies listed across the bottom, and there's a link to learn more about our collaborating labs. It's also important to mention that there's a full-featured PDF user manual that can help walk you through your first uploads, as well as how to dynamically pull data out of FishGen. So before a user can upload data to or export data from FishGen, they need to request an account. Upon requesting an account, FishGen administrators receive an email They can then verify the user's credentials and approve or deny their account. Unregistered users can still search for collections and save data sets, but they are unable to export any data. So let me give you a quick overview of how data flows into FishGen. First, a user has to upload their genetic markers into the database. A genetic marker is basically a very specific spot on one of a fish's chromosomes that, when investigated, it lets us maybe differentiate it from other fish. So one fish might have an A, the other might have a G, or one fish might have a T, the other might have a C. So you upload your lab's genetic markers to FishGen. Once your markers are uploaded, you can then upload collections and individuals along with various genetic data for those markers. We'll talk more about collections and individuals in a couple slides. So FishGen currently supports two types of genetic markers, microsatellites and SNPs, or single nucleotide polymorphisms. When we designed FishGen, one of the things we focused most on was standardization. Now, most of the problems that arose with previous online genetic repositories in the past were issues with data standardization. So when uploading genetic markers to FishGen, there are a number of safeguards and validation rules that prevent users from doing things like uploading the same genetic marker twice under different names, or uploading two completely different markers under the same name. A marker set is really just a group of genetic markers that are often used in tandem with each other. For example, our lab has a set of 96 genetic markers that we use specifically for our parentage-based tagging program. In addition, we have another 96 genetic markers that we use specifically for our genetic stock identification program. Each of these is considered a marker set. So users can choose to create a marker set at the time of marker upload, or they can just create one on the fly by using markers that already exist in FishGen. Marker sets can be used to export genetic data for various collections using a specific set of markers. They can also be used to view marker definitions for markers that were uploaded by other users into FishGen. So because of the data validation involved when uploading markers, when a user exports a list of marker definitions that may have been uploaded by multiple labs, the end data is standardized as far as layout and format. So let's talk a little bit about how collection and individual metadata are stored in the database. Now, in FishGen, individual fish are grouped into collections. Collection represents a group of fish sampled in a body of water either on a single day or over a period of time. So if you went electroshocking on a creek for 4 days and sampled 100 steelhead smolts, that's considered a collection. In FishGen, there are collection level fields such as body of water, latitude, longitude, etc. These are fields that apply to the whole collection. And then we've got individual level fields, things like marks, pit tag numbers, phenotypic sex, species, and so on. So once again, standardization of data becomes of paramount importance here. When people export data from FishGen, we don't want the format of that data to be different based on who it was uploaded by. So we accomplish this a few ways. One of these is required fields. Collections are required to have a latitude and longitude for GIS and mapping purposes, as well as either a body of water or a hatchery and a sample year. Individuals are also required to have an individual species. Another way we accomplish data standardization in FishGen is through format validated fields. If you pull individual data out from FishGen, you don't want to see a million different values in, say, the rear type field, things like W, hatchery, NOR, natural, hat, etc. The only valid entries for that field, therefore, are H, W, and U. So because of this, you can export collection and individual data that may have been uploaded by multiple agencies. You can export that out in a single file, and the data will be standardized throughout the whole export. So once data is into FishGen, we needed a user-friendly way to search for and export data. The search screen is one of two primary ways to find and export data from FishGen. When we designed the map search screen, we had user-friendliness in mind as our primary concern. So on the left, we've got an area where the user can specify search criteria, such as species, collection year, origin, hatchery versus wild, that sort of thing. There are also GIS-based fields that are calculated off of each collection's latitude and longitude, fields such as HUC, state, county. So the user can also select from multiple base maps over on the top right, which include things like satellite, topographic, and even a HUC layer. So once you search for collections on the map screen, a table of collections that met your search criteria appears under the map. You can then map this subset of collections on the map, or filter even further using additional fields. Once you've filtered down to the collections you're interested in, you can then export that data out in a handful of various formats, or you can create what's called a saved data set. So a saved data set is the other primary method of pulling data out of FishGen. A user can create a saved data set either when they upload a set of collections along with genetic data to FishGen, or by identifying a list of individuals and collections already in FishGen from which they'd like to create a saved data set. Other users can then browse saved data sets and export them using the marker set that was specified by the original creator of the saved data set. So the saved data set feature is invaluable to facilitating the easy sharing of data between labs. Rather than emailing large Excel files back and forth and worrying about things like versioning history and edits, you can just tell a collaborator to download a certain saved data set from FishGen. Saved data sets are also extremely useful for referencing data used in literature and publications. In a paper, you can cite that the data used for your study is available on fishgen.net under the saved data set named whatever you saved it as. Since a saved data set represents a snapshot of data that is immutable, anyone can then download that data set from FishGen and know that they're working with the exact same set of data that was used for a publication. So FishGen has been live for just over a year now, and it already houses some pretty impressive data sets. It currently contains 5 spawn years worth of hatchery broodstock individuals in the Snake River Basin for both Chinook salmon and steelhead. It also houses current genetic stock identification, or GSI baselines, for both Chinook salmon and steelhead in the entire Columbia River Basin. And lastly, I've included this Moyi Lake Burbot Parentage baseline to kind of highlight an important point. So although we originally intended FishGen to be used pretty specifically for PBT and GSI projects in the Columbia River Basin, hence the title of my talk, it quickly became apparent to us that it was capable of working for all sorts of other projects and species of fish. So we've actually broadened the scope of FishGen to include all sorts of projects across the Pacific Northwest, not just salmon and steelhead in the Columbia River Basin anymore. Our hope is that not only fish genetics labs, but also fisheries biologists and managers across the Pacific Northwest come to see FishGen as a valuable tool for the sharing of fish genetics data between agencies. So some basic acknowledgments. I'd like to thank Matt Campbell, our lab manager at the Eagle Fish Genetics Lab. FishGen was really Matt's brainchild from the start, and he's been really instrumental in the construction of the database since day one. Chris Kazefka, Craig Steele, Mike Ackerman, and Ning Vu are all biologists at our lab, and throughout the entire process have provided really valuable insight. Sean Nairn and Jeff Stevenson worked down at the Hagerman Genetics Lab with the Columbia River Intertribal Fisheries Commission, and they've been excellent consultants throughout the building of FishGen. Lastly, Bill Fleming, Chris Adam, and Mark Bricker are all employees at Resource Data Incorporated, who really worked closely with us throughout the entire project to ensure that the end result of FishGen was exactly what we envisioned. With that, I'll open it up to questions. I think. I'm not sure how that'll work if this is pre-recorded. Anyway, thanks for listening.